Welcome to Exploring with Scientists, How Have Some Frogs Avoided Extinction? Today is part 3, Working in the Field, and this is part 3 of a 4-part series. So one question that we have is, how have some frogs survived with a new disease, chytrid fungus, which we talked about in the previous session? So this frog here is one of the species that we study. This is called a harlequin frog. And what is special about this frog is it's something that we call rediscovered, which means it was thought to be gone and extinct and disappeared forever, but it actually wasn't, and it popped up again and was discovered again or rediscovered by scientists. So this is a frog that scientists were studying, and chytrid fungus arrived to this environment, and they all of a sudden kind of vanished. And scientists looked for them year after year, and as years went by and they didn't find any, we thought that they were completely gone and probably extinct. But when they popped up again, it created this really exciting opportunity to understand why, right? So they made it. They survived with this new disease, but why have they stuck around? How did they make it while other frogs did go extinct and did disappear? So this is one key question that we have that's related to this bigger question of how do some frogs avoid extinction? And to answer this question of how some frogs have survived with this new disease, we have to do a lot of things. And the first thing that we're going to talk about today, I'm going to walk through with you, is working in the field, doing something we call field work. So for this project, because I live in Michigan and work in Michigan, I travel from Michigan down to Ecuador, where the frogs live. And this is a country in South America. And I team up with fantastic scientists that are working down there. And we get together and come up with a plan. We figure out what we need, where we need to go, based on where using different maps and knowledge about where these frogs live. We gather all the gear and equipment we need to collect a sample and spend time out there in the field. We gather it all up, pack it up, and drive out to these actual field sites. So us kind of preparing for that is in the picture on the left, and on the right is us heading out to a stream that's kind of way out by that mountaintop you can see, where we think the frogs are living. And I wanted to show you a video of what it's actually like to walk through here. So I don't know if you can hear audio, but it's okay. It's mostly just to let you see, my view of walking through a tropical forest in Ecuador at one of these sites looking for these frogs. So this is a stream where we think they are living. So it's a small bridge that we can get across on either side to look for eggs or individuals near the stream that might be breeding. So this is a really beautiful place to work. In tropical forests, there are usually a lot of amphibians and reptiles. So in working here, we get to see a lot of cool different things that aren't part of, the questions that we're trying to answer. So when we go out herping, which is just going to look for amphibians and reptiles, these are two different kind of fun animals that we found. So on the left is a glass frog. And these are kind of popular to the public if you've watched any nature documentaries or things like that. They lay eggs on leaves that hang over rivers and when they hatch, eventually the tadpoles will hatch out and fall into the river. And the males, once the eggs are laid, will sit there and defend against like insects and things that try to eat them. And they also have a translucent underbelly, which means that you can see through. If you were to flip this frog over, you could see through its skin. You could see its heart beating, you could see it breathing, and all its organs in its body. So these are really cool to find. On the right, we have an anole lizard, and these are kind of famous for something that's called a dewlap. So there's another vocabulary term for you. And this is a patch of skin that they stretch out. It's a really cool pattern you can see. Different species have different patterns. And they use this to frighten other males or attract other females during mating season. And this is a really cool behavioral trait that they have. And there are some of these species even in Florida and in the US, different states, that'll be running along fence posts and things like that. And you can see them displaying these dewlaps. So I'm in the field and I'm collecting samples. What am I actually doing? I want to show you another video here. And you can see here that I have a frog in my hand, and it's actually the same individual. In the bottom left, this exact individual is the one that's in my hand in the video. And you can also see that I have a Q-tip. And what I'm doing is I'm taking the Q-tip and rubbing along its skin. So I discussed how the chytrid fungus is a skin disease. And so if we take the Q-tip and rub it along its skin, we can collect different things that are living on its skin. So we can collect the disease if it's there, and we can know whether or not the disease is present, and if the frog is infected with the disease. We can collect other microbes that are living on them, some that may be beneficial. And so that's what we're collecting in one sample. Another sample, which isn't part of this video, but is doing the thing with the Q-tip, is opening their mouth and collecting a buccal swab. So we're taking a smaller Q-tip, putting it in its mouth, getting saliva all over it, so that we can collect something we call the DNA sample, which I'll talk about later. And so this is what I'm actually doing every time we catch a frog. We collect these two different samples. We put them in a tube and label it and store it away, so we know which sample was for which individual. And then we release the frog. And so these frogs here and that I have on the slide are all frogs that we found and that we've collected these samples from. So that's what it's like to work in the field, doing field work with me. Thanks for joining for part 3.